But in addition to DNA, chromosomes also contain protein, and for the first half of the century, protein stole the show. A protein consists of an irregular sequence of smaller amino acids. There are 20 different amino acids which can be arranged into any possible combination. As the protein grows longer, the possibilities multiply astronomically. DNA, on the other hand, has an alphabet of only 4 letters, which were thought to be arranged in a monotonous sequence. It's no wonder that proteins seemed a better candidate to carry the language of heredity. It took a couple of scientific surprises for DNA to finally prevail as the stuff of the gene. The first took place in 1928 in the London lab of Frederick Griffith, a physician. He was studying 2 different strains of a bacteria called pneumococcus. One strain had an outer capsule and was highly infectious. When Griffith injected these potent bacteria into a lab mouse, it got sick and died. No surprise there. His next step was to kill these bacteria by heating them. When he injected the dead bacteria into a second mouse, nothing happened. Again, no surprise. Griffith's other strain of pneumococcus lacked the outer capsule of the first and were harmless. When he injected these bacteria into a third mouse, again as expected, they had no effect. Lastly, he combined the second strain with the killed version of the first strain. To his great surprise, this combination proved deadly. When Griffith examined the dead mouse, he found live bacteria that looked just like the infectious strain. This suggested to him that some factor had passed from the dead bacteria into the live ones to transform them. He also discovered that this transformation was maintained in all subsequent generations. So Griffith's transformation factor must have contained genetic material. Across the ocean in New York, a team of scientists at the Rockefeller Institute set out to identify Griffith's transformation factor. They were led by Canadian-born Oswald Avery, who, like Griffith, was a physician interested in the pneumococcus. Avery's team carefully separated out the chemical components in Griffith's dead bacteria and added them one at a time to living cultures of his benign bacteria. They found that the transformation factor was pure DNA. It was an astonishing result.